Kind of a quiet crowd here today. When you're tired from a busy weekend, rather be outside basking in the sun. We're going to wrap up some things having to do with circulation, which of course is related to gas exchange. We're not going to cover it in much detail beyond what is on your handout 10. And we're going to be referring extensively to handout 10 in today's class. So make sure you've got all pages of that available for quick reference. You kind of follow along and kind of dash through this stuff. I think the structure of vertebrate parts is very easily understood. Realize there are fundamentally two types of chambers in vertebrate parts, and this is right on your handout 10. So, just kind of follow along. The atrium is always a chamber that receives blood from the body, and then it simply pumps blood to its next door neighbor, the ventricle. Whereas the ventricles always pump blood away from the heart. Now, it should make intuitive sense to you that not much work should be required to simply pump blood through a valve, as opposed to pumping blood out through the body and through capillary beds, where there's more resistance to flow. So invariably, when you look at vertebrate parts, you should realize that the ventricles are much larger chambers. They're more muscular. They have to do more work. So that should make good intuitive sense. And then there's variation in how many of these chambers there are. You can find two chambers, and in two-chambered vertebrate parts, one is the atrium, one's the ventricle. This condition exists in lampreys as well as jawed fishes, except for lungfish. Three-chambered hearts consist of two atria and one ventricle, and this condition can be found in lungfish, amphibians, extant reptiles, except for crocodilians. And then four-chambered hearts have two atria, two ventricles, and this condition is found in us and all other extant mammals, as well as the clade that includes crocodilians and birds, and very probably dinosaurs. Obviously, there's some problems trying to understand what soft tissue is based on fossils. But given this distribution in extant taxa, it seems a pretty good bet that the most recent common ancestor of crocodilians and birds also had four-chambered hearts, as that would be the most parsimonious explanation. Now, when you're considering circulatory systems, just one other thing to keep in mind is that arteries are blood vessels that are involved in moving the blood away from the heart to a capillary bed. Whereas veins are blood vessels that are receiving blood from a capillary bed and returning it to the heart. A common misunderstanding is to assume that blood is oxygen rich in arteries and oxygen poor in veins. That's true for many arteries and many veins, but not all arteries and all veins, because of the circuit that can actually send blood to the gas exchange surfaces and therefore oxygenate the blood. Now, you've got this slide on your handout, and I apologize for its poor quality. However, if you go to the Canvas site, you've got this in color, and it is actually legible. So I wouldn't be overly worried about scribbling all this stuff down since you have that on the Canvas site. So this is just an overview. If you were to follow a red blood cell starting at the atrium in a fish, of course, it would go to the ventricle. And then the ventricle pumps blood out to the gills, where there's a capillary bed, and oxygen is picked up So that's why I'm showing this in red. And then the blood is conveyed to the non-respiratory tissues, where there would be additional capillary beds, and then returns to the atrium. Lungfish are different. They're still fish, but they have 3-chambered hearts. So they have 2 atria, and invariably it is the right atrium, when there are 2 of them, that receives the blood from the body, passes it off to the ventricle. Ventricle pumps blood to a combination of the gills and the lungs, where it's oxygenated. And then this comes back into the heart via the left atrium, and then leaves the heart via the ventricle. Okay? And then from the ventricle, it's going to non-respiratory tissues, and then ultimately it returns to the heart. Okay? And in amphibians, not much is too radically different, except you don't have gills in the mix. So you've got going from the right atrium to the ventricle, to the lungs, left atrium to the ventricle. And then, interestingly, depending on the species of amphibian, some of them, many of them, actually have capillary beds in the skin that pick up oxygen. So there are 2 important sites for gaining oxygen in adult amphibians, namely the lungs, and in a great many species, the skin. And the relative importance of those 2 varies tremendously among different species of amphibians. And I trust you're aware that a very large group of, on average, very little salamanders are lungless, and that family of salamanders is what? What's the scientific name of the family of lungless salamanders? You've had your amphibian lab. What's the most speciose family of salamanders? There's one family that has the vast majority of living species, and that family is what? Plethodontic. Plethodontic, also known as the lungless salamanders, and a great many of them are very modest size. A great many of them are also quite skinny for their mass. And of course, the more you depart from a spherical shape, the greater your ratio of surface area to mass. Okay? So, simply by virtue of small size and their shape, they've got some pretty viable options for using the skin as an exchange surface exclusively. And then in 4-chambered hearts, again, blood is always coming to the heart via the right atrium. And then in this case, you've got 2 ventricles. So you go from the right atrium. You always go from the same side atrium to the same side ventricle. And then you have blood going from the heart to the lungs. And realize that when the blood is going away from the heart, at this point, it's oxygen poor blood. And then when it hits the lungs, it picks up oxygen. That's the pulmonary artery. So that's one that can take you out if you fall into this trap of thinking of oxygen-rich versus oxygen-poor blood, defining what veins and arteries are. Instead, the most generic definition is simply blood's going from the heart to a capillary bed or from capillary bed to the heart. Okay? And then after getting oxygenated in the lungs, returns to the heart via the left atrium and the left ventricle. And then the left ventricle does the heavy lifting of sending the blood to the vast majority of the non-respiratory tissues in the body. And that is a lot more stuff than is present in the capillary beds of the lungs. So if you see a heart in lab, and that heart is actually a four-chambered heart and it's out of the animal, it's pretty easy to determine left and right sides because the left ventricle is bigger than the right ventricle, and it commonly makes up the apex of the heart. So once again, we'll see some nice examples of structure-function relationships. If you need a bigger, stronger pump, you have more muscle and you have a bigger chamber in the heart. Okay? So that's a quickie overview. Again, be confident about putting the evolution of vertebrate parts onto a phylogeny to explain this in the most parsimonious fashion. Okay? I like this picture that shows the Rio Negro. Negro means black. So that's the dark water river entering the Amazon, which has very muddy water. And beyond the fact that that's kind of a cool picture, is that at all relevant for patterns of circulation in vertebrates? Who says yes? Raise your hands. Who says no? Raise your hand. And then we got some people just, who knows. Okay, but a lot of you did say yes, so then why?
Because it's different things in the body, the smaller blood vessels and capillaries merge with the larger ones.
I'm thinking of something different. What's an example in the vertebrate circulatory system where you've got two big vessels dumping into a common space?
Is it like the oxygenated and deoxygenated blood going into the heart?
What kind of heart?
Would it be the two-chamber?
Let's have a bidding war. Let's go up to 3 chambers. Okay? The reason I show this is just because you do not have 2 separate chambers, or for that matter, you realize that some taxa have a partially divided ventricle rather than 2 separate ventricles. Patterns of flow can be very complicated. And as illustrated by these 2 rivers meeting, there can actually be, over a limited distance, a rather modest amount of mixing. So sort of the old view of a 3-chambered heart was, you've got oxygen-poor blood meeting oxygen-rich blood, and then it all gets stirred up in the ventricle, and then it goes out and it's kind of halfway, It's actually not that simple. Okay, so be forewarned that you can still have a very modest effect of decreasing the oxygen content in blood when you have deoxygenated blood and oxygenated blood meeting in a common ventricle. But exactly how much depends on a whole bunch of gory details. But watch out for that, okay? Because kind of the old view of a 3-chambered heart is that there's just a complete mixing. And that's pretty much become an antiquated view, okay? Well, let's move on to something different. Namely, how is energy acquired? Well, amphibians eat stuff. Of course, there's heterotrophs, uniformly heterotrophs. And there are a number of different ways that amphibians do eat. And part of what you should master is the role of the hyolingual apparatus. What moves and what muscles are used. And for both Neuron amphibians and for salamanders. Okay? Now remember, we've already seen that the hydrobranchial skeleton in neurons especially but also, although we haven't talked about it, to some extent in adult amphibians, serves an important role for ventilation. But as we're about to see, it also is involved in feeding. Can you think of another group of vertebrates where the hyoid and branchial elements have those two functions? Is this unique to amphibians? No. Well, then, what makes it not unique to amphibians?
Maybe fishes.
Yeah, okay, in fishes. I think it's kind of interesting. These are two rather different functions, right? Eating and ventilating. And depending on some of the details, you may actually have the potential for a functional conflict. So in theory, you might get this apparatus so modified that maybe it's good for feeding, but it's not so good for ventilation, or perhaps the other way around. Okay, so that's something to think about. Okay? And salamanders, especially, have been studied for both their terrestrial and aquatic feeding. And for adults, of course, a whole bunch of amphibians have aquatic larvae, but some of the best studied aquatic feeding of amphibians is for adult salamanders in water. So on page 10, you have these anatomical diagrams of some of the relevant muscles, some of which we've already discussed with respect to feeding. What are three key muscles that we have already discussed with regards to ventilation in a neuron amphibians? Someone in the back row, give me one. Back row, this side of the room.
Genealogist Muscle.
Very good, the geneal hyoid, and in the next row up, the geneal hyoid goes from where to where? Second row from the back, this right side of the room. Remember, that's an origin insertion name. So break that apart to genial and hyoid. Next row up, can you help out?
Isn't it give from the mandible to the hyoid?
Mm-hmm. And put another way, I find it as a memory aid easier to say jaw, because jaw is kind of like genial. You get that same sound, right? Okay. And then next row up, give me another muscle. It's very important for ventilation in the neuron amphibians.
The sternum hyoid.
The sternum hyoid, yep. So that's going here. And you two, that goes from where to where? Sterno.
Just from the sternum to the hyoid.
Yeah, okay. And in this case, the pectoral girdle. Okay, so the sternum is part of the pectoral apparatus, if you will. And then someone in the front three folks over here, give me another muscle that's involved in ventilation. You got the geniohyoid and the sternohyoid. What else? And I'll give you a hint. It's in that middle picture. So if you skin a frog, that's the most superficial muscle under the lower jaw. Another name for the lower jaw is the mandible, and it goes from left to right side on the mandible, so it is in between, and therefore it is the intermandibularis. Okay, so that goes from left to right sides of the lower jaw. That makes that sheet of muscle. Remember that the straightest or the shortest distance between 2 points is a straight line. So if here are the left and the right sides of a jaw, and if you have a bulge downward, that's a longer distance than going straight across. So if you have fibers that go from left to right, they're going to shorten, okay, and make that flat. That would minimize their distance, okay? So this stuff is basically involved in raising and lowering the floor of the buccal cavity, which creates those changes in pressure, positive and negative pressure. Negative pressure draws air into the buccal cavity. Positive pressure pushes air out of the buccal cavity into the lungs. So that's what's going on with respect to ventilation. But these things also have a role in feeding. In addition, there's this little muscle with fibers that go from left to right, and it goes from the left to the right half of the lower jaw in the Neuron amphibians. And in the Neuron amphibians, the lower jaw is kind of bendy compared to our lower jaw, okay? And then there's some additional muscles. Glossus refers to tongue. Once again, genial refers to the lower jaw, and the genealoglossus goes from, in this cross section, this would be the lower jaw, and then you've got fibers for the tongue at rest, where the muscle actually makes up the bulk of the tongue. So tongues are muscular structures. And the other muscle that makes up the bulk of the mass of the tongue, when the tongue is at rest, it's on the ventral side of the tongue, and then it wraps around the hydrobranchial skeleton. So none too cleverly, that's referred to as the hyoglossus muscle. So it originates on the hyoid, it inserts into the tongue. This originates on the tips of the lower jaw, and then inserts into the tongue. So I know that some of these names seem a little intimidating, but if you remember the meaning of these root words, these are some pretty simple ones to remember because they're actually telling you where the two ends of the muscles are attached. These are mostly origin insertion names. And realize that the mental region is a synonym for the chin region of critters. Okay? So, what happens when a neuron amphibians feed? You have these illustrations on handout 10. These are cross sections for an neuron amphibian feeding. And this is showing the tongue at rest. This is showing the tongue fully extended.
We booked a cozy Virgo mountain cabin with a kitchen where everyone can chef. Let me know number for help.
And this is a beautiful slow motion video of a southern toad feeding. Notice how the lower jaw is curved. The tongue is flipped out of the mouth. It's really a drag that we have our masks on, because when we don't, I tell everyone to stick their tongue out at me. But do a thought experiment. When you stick your tongue out at somebody, what happens is simply that the tip of your tongue goes farther out. Okay? When we stick our tongues out, the top side of our tongue remains the top side, and the bottom side remains the bottom side. That is not how neuron amphibians shoot the tongue out of their mouth. An analogy for what's going on in a neuron amphibian, not only can you use your arms to imitate Sammy Salamander, you can use your arms to imitate the action of a frog tongue, where this would be the dorsal surface of the tongue at rest. It would be wrapped around the hydrobranchial skeleton. and it flips out like this. So when it hits the prey, the dorsal surface at rest is oriented ventrally and vice versa, okay? That's fundamentally different than the way we stick our tongues out. So the other thing to keep in mind is when the tip of that tongue is extended, that portion of the tongue, when it's in the mouth, is the most posterior part of the tongue. So it flips out. It does not simply extend out like our tongues do. So a very odd movement compared to the way our tongues work. So how does that happen? Well, electromyography has been used to document when the muscles are active, and I'm skipping some of the details, and just these are sort of painted with the broad brushstrokes in the highlight, but there's early activity of the Genealogist muscle. Now remember, that's the muscle along here in this diagram. This is the lower jaw, and this bigger round structure in cross-section, that's the submentalis muscle. And then the Hyalurosis muscle is here, okay? So envision this muscle is active, and that does two things. That makes the tongue stiffer, but then those fibers shorten. And since those fibers are oriented lengthwise, as they shorten, that starts pulling the back part of the tongue forward to the lower jaw, okay? But then another thing that happens, if you look closely at this diagram, you'll notice that the orientation of the submentalis muscle relative to this cross-section through the lower jaw shifts. Namely, transiently, that submentalis muscle creates a bulge above the tips of the lower jaw. But the tongue must get around that. So one thing that's presumed is this helps impart some upward movement to this motion and creates something of a pivotal point about which the tongue moves. So a couple of things that this muscle does is it depresses this symphysis between the left and right halves of the lower jaw. It lowers it. Remember how that lower jaw looked curved in the feeding toad? And then also it's believed to create something of a pivotal point about which the tongue can flip. Okay? And then as time goes on, there's continued activity of the Genealogist muscle. And that proceeds to protrude and flip the tongue out of the mouth. And as I mentioned before, ultimately, it's the dorsal surface at rest actually hits the prey. And there's a bunch of sticky stuff on the tongue that helps in this process. And then later on, we see Hyalurosis muscle activity. Now remember, the Hyalurosis muscle, when the tongue is at rest, it's making the ventral surface of the tongue. And notice how when the tongue flips out of the mouth, that muscle gets stretched. So then if that muscle shortens, that's going to start bringing the tip of the tongue back towards the mouth. Namely, it retracts the tongue. Because of the way the fibers actually fan out in the tongue, that sort of helps make a cup-like thing that envelops the prey. And then when the tongue is being retracted, there's some Sternum Hyoid activity. So envision something tugging on, and it's not shown in this diagram, but the straight line represents the Hydrobranchial skeleton. So of course the Sternum Hyoid can tug this structure this way, okay? And it can also, even if that structure doesn't move, stabilize that structure while these other muscles that go from the hyoid to the tongue are active. Okay? Notice that curvature of the lower jaw. Okay, just barely gets the cricket. And then another little fun fact, although it's not shown in that video, is swallowing in the Neuron amphibians is almost always facilitated by closing the eyes. You've seen frog skulls in lab, right? And there are two huge spaces where the eyes go. So then there's a bunch of soft tissue around where the eyes are. So if the eyes get pushed in, they've got to go someplace, and they can actually help push prey in preparation for swallowing. If we close our eyes when we swallow, it doesn't do a darn thing other than closing our eyes. So again, different structures, different functions. Well, now let's consider some highlights of how salamanders feed. And of course, some salamanders are aquatic. And if you have an aquatic salamander, what do you think might be a way that the critter could eat? And mind you, they're capturing animals. All salamanders are predatory, visible.
Suction feeding.
Okay. Indeed, there are salamanders that use suction to capture prey. So it shouldn't be too surprising that there are all kinds of similarities in the movements and the mechanics involved in creating negative pressure and sucking water plus prey items into the mouth. Okay? Now remember that when fishes use the suction pump, either for ventilation or for feeding, water enters the mouth and it exits the opercular valve. So you've got nice flow from front to back and just in one direction. But in order to have that unidirectional flow, because of the way other structures are built, you need a separate entrance and a separate exit in order to have unidirectional flow. Well, larval salamanders still retain pharyngeal slits, okay? Even though there's no opercular opening, there is in fact pharyngeal slits associated with the gill arches. So you've got something that can function as an exit when the mouth is functioning as the entrance for sucking the water in, okay? But interestingly, there are some species of salamanders that effectively lack a sizable exit for the water, and yet they still use suction feeding. Well, that water's got to go someplace, and salamanders don't want to get a big tummy ache from a big gulp of water. So in fact, the water goes in the mouth, but is also expelled from the mouth. Okay? Of course, there's a lot of terrestrial feeding that occurs in salamanders. And once again, we see that the tongue plays a vital role If the tip of the tongue is extended beyond the tips of the jaws, that's referred to as lingual protrusion. Now you're aware that the tongue is associated with the hyoid skeleton. So there are a bunch of muscles of the tongue that are attached to different parts of the hyoid apparatus. And interestingly, in some salamanders, not only does the tongue go beyond the tips of the jaws, but the hydrobranchial skeleton also goes beyond the tips of the jaws. So lacking imagination, biologists refer to that as hyolingual protrusion. Okay? Because the hyoid, as well as the tongue, is going beyond the tips of the jaws. And this type of movement is best developed in Plethodontics. Remarkably, in some Plethodontics, as shown in your book, the tongue can be extended more than half the body length of the salamander beyond the tips of the jaws. So that tongue is actually the primary capture mechanism. And then when the prey are brought back towards the mouth, the jaws may clamp down and hold the prey and kind of mush the prey up a little bit before being swallowed. Why don't we see this in other groups of salamanders, do you think? Or why do you think we see it in such a spectacular fashion in the plethodontic salamander? In many amphibians, the hydrobranchial apparatus has two functions, and those two functions are
respiration and breathing, right? Eating and feeding.
breathing's not the best choice of words, because that includes a bunch of stuff like gas, ventilation, and feeding. Okay? And what's different about plethodontics?
They don't have a lot of water.
Oh, so they don't need to pump air into the lungs. Okay. So a very plausible explanation for this highly specialized anatomy in plethodontics that allows this tongue to be shot way out could be that since they no longer have a reliance on the structure serving as a buccal pump, its form can be radically modified and do something else, right? Because it's not constrained by the need to also function as a buccal pump. Okay, and you've got these figures on your handout 10. These are ventral views of the hydrobranchial skeleton of a plethodontic salamander. And if we go from anterior to posterior, this unpaired skeletal element is the basibranchial. And then behind the basibranchial are the ceratobranchial. And then these very long and slender parts at the end of the line are the epibranchial. And then below here, we have some of the key muscles for understanding hyolingual protrusion. Remember that antagonistic muscles are muscles that have opposite functions. So if you activate your biceps muscle, That flexes your elbow. And if you actively extend your arm, you're using your triceps muscle on the other side of the joint to increase the angle of that joint. But what about these muscles? Well, the subarcualis rectus, and I can't think of a handy memory aid on that one, so it's kind of stuck, has fibers that run from anterior to posterior, okay? So this attaches to the basibranchial anteriorly and goes all the way back to the chest region in salamanders, and plethodontic salamander. Okay? Oops, I'm sorry, I messed that up. That is the rectus cervicus artery that is oriented longitudinally. Whereas the subarcualis rectus is this one that wraps around the epibranchial bone. Okay? I think this is pretty interesting because these two muscles have opposite functions, but they're not on opposite sides of a joint. And to a close approximation, not only that, but the fibers of one are at a right angle to the fibers of the other. So what the heck goes on in these crazy salamanders that shoot the tongue and the hydrobranchial skeleton out of the mouth? Well, the subarcualis rectus, whose fibers are oriented circumferentially, when those fibers shorten, it's just like me squeezing my fingers. And you'll notice that there's a taper on that epibranchial. So if you squeeze on something, the tapered end is going to go away from being squeezed, okay? Because the tapered end is smaller, okay? So envision this muscle wrapped all along here, and it pushes that long skinny bone out of this tube made by both the muscle and connective tissue. Bringing the tongue back into the mouth is rather straightforward because you have this other muscle with fibers that are oriented longitudinally, and they simply shorten and bring everything back in. Now, during hyolingual protrusion, I want to emphasize the orientation of these bones change. As they are pushed out of the mouth, because the way the connective tissue is arranged, instead of staying like a wishbone, the ends come together, and you basically get a rod-like structure as it's pushed out of that sheath made by the subarcualis rectus. So you get a long, skinny thing, okay? So let's look at some examples of some of these different ways that salamanders eat. You're well aware, I hope, after lab, that Cryptobranchus is kind of the basal group of salamanders. Highly aquatic as adults. Sadly, they're quite sensitive to water quality, so in a lot of parts of their range, they're kind of hurting from pollution. That's not the hydrobranchial skeleton in the background. But here we go. Now, this obviously is not a prey item, but it still illustrates suction occurring. So remember, the key criterion for suction is, does the animal move something towards it by using the water, as opposed to the predator moving to the thing, okay? And here's a little air bubble that is about to get sucked into. Look at that tremendous expansion of the oral cavity, and you can see some of the hyoidobranchial skeleton here that is involved in that. Okay? So notice that tremendous increase in volume that causes a big decrease in pressure. But then ultimately, these guys need to expel the water from the mouth. Okay? Now, there is actually a pharyngeal slit in Cryptobranchus, but it's so small that it is not sufficient to expel all of the water. So they would be an example of using bidirectional suction feeding. But of course, many salamanders are up on land, including many ambystomatids, the tiger salamanders and spotted salamanders. Okay, and there you can see the tongue goes a little way out of the mouth. It's sticky, and the tongue draws the prey item back into the mouth. Eurycia is one of the genera Plethodontic salamanders that you've seen in lab. It's a North American group. It has a moderate amount of hyolingual protrusion. These are slowed down tremendously. A lot of these events are happening on the order of about 1/50th to 1/30th of a second, faster than the blink of a human eye. Okay? So that tongue goes a good long way compared to ambystoma, and part of the reason for that is that the hydrobranchial skeleton is actually going beyond the tips of the jaws. And then perhaps the all-time champion in this department for distance in the genus Hydrobranchial. Look at that tongue shooting out of the mouth. Now notice one thing that I really like about this video, you can see this little discontinuity. See the little bulbs? That's where the tip of some of those hydrobranchial bones are. So notice how this is rather straight compared to this is more irregular. Okay? Yummy. If all of you have lunch, probably not eating crickets, although you could. I think insects are reasonably nutritious.
So those bones go all the way out to that point that you pointed at?
The anterior portion of those bones were going to that point you could see where there was a little vent here. So they're going well beyond. It's important to realize that even with just lingual protrusion, that the hydrobranchial skeleton moves some, but the key break for calling it hyolingual protrusion is does the hyoid apparatus go beyond the tips of the jaws? So it can wiggle around down here without being hyolingual protrusion, but in these crazy plethodontic salamanders, it actually goes beyond the tips of the jaws. And you're probably well aware that temperature has profound effects on physiological processes, including the rate of force development. And of course, amphibians are ectotherms. Ectotherms depend on environmental temperatures, environmental heat sources to attain a particular body temperature. And a lot of salamanders are actually nocturnal. They're out and about when it's rather cool. Potentially, this poses a dilemma. So you would sort of expect then, if mussels were just doing their normal thing, that when a salamander's cold, the tongue is going to move more slowly than when a salamander's warm. But there's some additional little tricks that salamanders have up their sleeves. Well, I guess I've never seen a salamander in a shirt, but you get the idea. Namely, it appears, okay, so here we have the salamander at a body temperature twice as high as this. This is greatly slowed down, and for all practical purposes, the hyolingual protrusion is an identical speed, despite a doubling of body temperature. But tongue retraction is not. So, that rectus cervicus muscle that just has straight fibers is kind of doing a normal muscle thing. and it can't generate force as quickly when it's cold. The implication of this temperature independence is that there's actually an elastic storage mechanism that facilitates those very high accelerations. Because it's actually physiologically impossible to account for this by what the muscle alone is doing. The details of exactly how that elastic mechanism is triggered are still not completely well understood. Okay? So lots of different structures, and different structures can function in different ways. Now, before we leave feeding of amphibians, please remember most, not all, but the vast majority of amphibians have larval stages that undergo metamorphosis, and the vast majority of those larval stages are aquatic. And if you're aquatic, you have very different constraints on how you can feed compared to whether you're terrestrial. We talked briefly about some of the anatomical correlates of tadpoles associated with whether they're in still water, flowing water, transiently terrestrial, or whatever the case may be. You also have correlates between the morphology of the mouth parts of tadpoles and what they eat. Now, a lot of tadpoles sort of scrape things off, like algae and so on, various films. But there's some tadpoles that are actually carnivorous, and they have pretty well-developed beaks. Now realize, these are not teeth. These are just keratinized structure, just like the so-called claws on clawed frogs is just keratin. That's the same stuff that's in our fingernails. So it's a hard, horny substance. But you can see at a glance, depending on where the tadpole is located, you may have a different mouth orientation, you may have different mouth parts, and so on. Okay? And I think in one of the more intriguing strategies, okay, here is a cannibalistic spade-foot toad tadpole. Look at this monstrous thing. This is another tadpole from the same species, but it is not the cannibal morph. Spade-foot toads are found in many areas in the arid southwest where the water evaporates very quickly, so there's a premium on metamorphosing very rapidly. Eating things like basically pond scum and algae is a very poor quality diet compared to actually eating something that has all the same constituents that your body has. So these cannibal morphs not only are bigger, they have different mouth parts that allow them to munch on these poor little guys that are not cannibal morphs. So obviously, tadpoles have profound differences in morphology compared to adult and neuron amphibians. Now, even though tadpoles are aquatic, suction feeding is, as a predator, is really rather uncommon. There are some groups where it occurs, and it isn't well studied. But interestingly, that is a pipid tadpole. And even though this is slow motion, it's doing it so fast. So here's a little prey item. Here's the tadpole. Remember that acid test for whether or not suction feeding occurs is what moves relative to what. Effectively no movement of the predator, and the prey is sucked into the mouth. Okay? But that is sort of an exception to the rule. Okay, that wraps up things having to do with how amphibians eat. Please remember your next take-home assignment is due beginning of class on Wednesday, so be sure to get that polished off. And as always, continue to read ahead on the handout to the next collection topic, which I think you'll find fascinating, namely reproduction in amphibians.